there are huge ethical and cost issues associated with using animals as model systems. Now, in the context of infection biology, it adds another layer of problems because we have to infect the animal. So, in terms of the kind of facilities required in the animal house, the stringent precautions one will need to take. And often you don't get those permissions. So that's one aspect of it. The other aspect of it is when you look specifically at wound biofilms or specifically at oral biofilms, animal models are not really the best mimic for the kind of infection we see in humans for different anatomical and physiological differences that exist. We don't think animal models are they are not to be, we wouldn't say that we are in a place where we think we should completely replace them, but we can certainly reduce their use. And one way of doing that is by building human relevant in vitro systems that can sort of in the drug development pipeline serve as a step before you go to animal models.
Hey, hi, welcome to Biotech Talks. Today with us we have Dr. Karishma Kaushik, Assistant Professor at Institute of Bioinformatics and Biotechnology, Pune University. She is developing human-relevant in vitro systems with an attempt to replace the model organisms in the drug development process. Now, why and how she is doing all these experiments and what other experiments are going in her lab will be explained by her in this episode of Biotech Talks. I hope you enjoy it. Hello ma'am, how are you?
Doing very well. Thank you for this opportunity.
Yeah, and thank you for your valuable time. So, your lab is known as Human Relevant Infection Biology Group.
Right.
So, why is that name?
So most faculty or principal investigators sort of name their lab after their name. But I wanted to give it a descriptive title that would convey to people what we do. So I'll tell you the backstory behind it later, shortly, but to summarize in one line, it's basically because we study infections in contexts that are relevant to humans. So we all know that bacteria cause infections, for example, but standard microbiology has always focused on studying them in tubes, in vessels, in wells, in chambers, where they're attached to, say, polystyrene surfaces or in the presence of lab media, which is rich in protein and artificial proteins like, tryptophan, extracts, etc. But we want to move beyond that and want to move to a space where we study them in the presence of factors that are there in the human body. For example, the serum proteins that are present, or attachment to surfaces such as collagen, where bacteria would likely attach in human infections. And so we study infections in that space where we bridge the human body and the microbe. But we do it in a lab, so we do it in vitro, and therefore we call it the Human Relevant Infection Biology Group. So the fun backstory behind this is when I started my lab in 2018 at Savitribai Phule Pune University, where we are right now, one of the first things I decided to do was make a Google website for my lab, because I said I've returned to India, I know nobody knows me, and the only way anyone can search our group and search about what kind of work we do would be to have some web presence, to have some website. And there I had to buy a Google domain, And I remember thinking in 2018 that I will leave it as karishmakaushik.lab.com and not Human Relevant Infection Biology, because I don't know where exactly my research will lead me. Right now I'm focused on, say, wound biofilms, but over the course of the years we moved to oral biofilms. We moved to biofilms in different contexts. I remember thinking that the lab name may change, but my name is not likely to change, at least less likely to change in comparison to the lab name. So we christened the lab Human Relevant Infection Biology, but our web link still has my name. And that's the story behind that. I remember thinking that, oh God, it's going to be a pain to change the website link once you've shared it with everybody.
Yeah. So you study animal models, and
right.
The problem with animal models, that is what I read about on your website. So what is the problem with animal models?
So there are huge ethical and cost issues associated with studying in vivo studies, associated with using animals as model systems. Now, in the context of infection biology, it adds another layer of problems because we have to infect the animal. So in terms of the kind of facilities required in the animal house, the stringent precautions one will need to take. and often you don't get those permissions. So that's one aspect of it. The other aspect of it is when you look specifically at wound biofilms or specifically at oral biofilms, animal models are not really the best mimic for the kind of infection we see in humans for different anatomical and physiological differences that exist. And we don't think animal models are uh, they are not to be, we wouldn't say that we are in a place where we think we should completely replace them, but we can certainly reduce their use. And one way of doing that is by building human relevant in vitro systems that can sort of in the drug development pipeline serve as a step before you go to animal models. So like they will be bridging the gap between very, very simple lab models, which is like bacteria in a tube, versus, advanced or so-called advanced animal models which are not really human relevant. So we think the in vitro models we build can kind of bridge that space. And sort of if you think of, you have thousands of molecules that you're evaluating to be antibiofilm candidates. Lab results will be great because there's no serum to blunt the effect of those antimicrobial peptides or small molecules. And then we take them to animals and invest so much time and resources in testing them in animals only to realize they're not working. And so we're basically stuck there. What models that are human relevant but still in vitro can do is allow you to relatively high throughput manner evaluate these several molecules and choose ones you take further for testing, either to animal models or advanced in vitro systems, or then into the clinic.
Yeah. So Dr. Abhijit Majumdar
Yeah,
at IIT
yeah,
Bombay is
he does.
developing organoids.
Yes, he does similar work. I don't know if he's doing it in the infection space, but we are sort of, both of us overlap in regions of like lab on chip, microfluidics, in vitro systems, but yet for some reason we haven't been formal collaborators. So I don't know, Abhijit, maybe that's a call that we need to do some collaboration.
Yeah.
Who knows? Yeah.
So what is your approach? Like how you are developing that in
So,
vitro system?
so like I said, we use in vitro platforms. Like our platforms are still 96 well plates. They are 6 well plates. They may be lab on chip systems. Or we do things like we coat them with collagen or coat them with fibrinogen and fibronectin, which is something that you find in wounds, say, for example. Then we build milieus or media, which are a combination of different proteins, biochemical factors, and matrix elements. We grow bacteria in them. Sometimes we grow bacteria in them in these systems. So we're replacing the milieu or the microenvironment of the infection state with human relevant components.
Okay. So is there any example like?
So for example, we've developed an in vitro wound milieu. We call it in vitro wound milieu or IVWM. When people study wound infections, they study the common pathogens like Pseudomonas and Staph aureus, but they study them in LB. They study them in broths, lab broths. We looked at clinical composition of wound fluid. This was based on a lot of publications that happened in nursing journals where people had sampled wound fluid and enlisted its biochemical and chemical components. And so this is not work we did. We used previously published work, which was a lot of clinical and nursing publications, and we recapitulated that formulation like making a recipe in the lab. Like, all right, if it contains so much albumin, how much do we need to add to get that kind of concentration in our in vitro wound medium? So that's the kind of work we do. We built this platform out, this medium out. And then we found that it could support the growth of biofilms. In biofilm forming pathogens, Pseudomonas and Staph. We could recapitulate several important clinical features like antibiotic tolerance, metabolic activity of biofilms, which basically says, again, this on its own, even if we don't put it in the context of a lab-on-chip system, this medium on its own can serve as a preclinical screening. So in addition to evaluating a small molecule or antimicrobial peptide in LB, which would be like really the first pass thing, that's the first thing you would do, you could also evaluate it in the wound milieu. And if it does have an effect, then we could probably think of taking it further. Because if it's going to have any effect in the human body or in the animal system, it's going to have to be effective in the presence of serum, matrix elements, which are very likely going to influence the effect of the potential antibiofilm agent.
Okay.
So we can use that milieu or medium that we've developed as a standalone preclinical tool. We can also use it with cellular elements in more complex systems as preclinical screening tools. And that's the work we took further, which is now a preprint, where we took the milieu, we had already characterized it and published it, but now we put it with cells that are normally present in the wound bed, which are like human dermal fibroblasts and epidermal keratinocytes. So now it becomes like we've called it a 4D microenvironment, where the fourth dimension is not time, but the fourth dimension is this biochemical nutrient milieu. So if you think of the wound bed, there'll be cells. So we can grow these cells in a like say a 6 well or 96 well plate. And then the fourth dimension will be this biochemical profile. And we've characterized the structure of Pseudomonas and Staph aureus biofilms in this environment. So it's currently under review. The preprint is out, it's under review. So we are waiting to see how the community responds to it. But these are the kind of simple and then stepwise advanced systems that we aim to develop. Now, the simplest system was the milieu or the medium. The second step would be the cells. The third step would be we have no biophysical factors as yet in our systems. So we can add factors like shear, fluid flow, tension and compression forces, and see how they influence the formation of biofilms, but in the presence of all these factors. So we are trying to get closer and closer to the human wound biofilm state.
Yeah, as a biotechnology student, I only realized this recently while talking with Dr. Abhijit Majumdar that there are these physical forces which play very important role how a cell behaves. And for me, it was very surprising because all I knew is there are these genes which control the cellular functions and that's all. But these physical forces that you are doing research on and Dr. Abhijit lab is doing research on are really interesting things.
I know we often think of the cell as very governed by within, right? Yeah. We don't realize, or we sometimes miss the picture that the cells are actually influenced heavily from outside. And any biofilm that forms on these cells will have the same factors, it will be subject to the same factors. It's very interesting to think about it because when we treat biofilms in wounds, one is we try and bombard it with antibiotics. We still don't know why they fail, why they succeed. Most often than not, they don't work, requiring very long-term and repeat usage of antibiotic. But another thing people do is mechanical debridement of the wound, surgical debridement. And understanding the physical forces at play may help us build better debridement approaches and strategies, which we can use in conjunction with antibiotics and peptides and other agents.
So I saw your Twitter profile and there is this AMR background picture that
the AMP actually. AMP, that's our antimicrobial peptide repository. Okay. That's another piece of work that we did last year. This was actually a very interesting project. It was a fun actually project because it was a remote internship. I worked with 4 young people who were in Bombay, between Bombay and Pune. They just emailed me during the pandemic, and I think this would be familiar to many young people watching and listening to this, that there were no internships possible for the 2 years. There was no way people could host anyone in their lab when they themselves were not in the lab. And when these 4 students emailed me, I said, the best I can offer you is I have this idea. We haven't yet had a dedicated repository of antimicrobial peptides for biofilms, which focuses on, the 3D structures of the peptides, annotations to relevant literature, where somebody says, all right, I want to look at developing novel antibiofilm approaches. What's the one-stop place that I can go which will have resources? AMPs are an excellent candidate to explore, especially because for wound biofilms, you can apply them topically. And so you kind of bypass a lot of the issues with pharmacokinetic availability, etc. if you had to consume them, provide them through the oral route, when you think of it as a topical application. And so then they said, yes, we are ready to work on it. And we, over the course of 1 year, built this massive repository of 5,000 antimicrobial peptide structures and a vast library of over 11,000 annotations using all publicly available information, like databases that were already in existence, like PubMed, for example, or the DRAMP database for antimicrobial peptides. But this is a one-stop repository dedicated to sourcing and searching for AMPs for biofilm studies. So that is that header on Twitter. That is that header. In fact, we finished that, we've published it. Now we are on the second leg of that project where we are not looking at the peptides. We looked at the peptides already, the database is already there. We're now looking at the target side. Okay, you have thousands and thousands of peptides, but we also need to see what target in the bacteria those peptides would interfere with or inhibit. And so our next step is we are now working on writing the manuscript for the target side. So we're sort of looking at the other side. But I think I won't say too much because it's not yet a preprint and it's still in the writing stage. But yeah, what's very exciting, I would say since 2018 to today is that we've published the first set of papers in all our projects. Now we are in that exciting phase where we are publishing the second set of papers that builds on previous work. So for a young research group, when you get to the place where you're building on your own previous work, it's always very exciting. Yeah, and so several of our projects are in a stage where we are now into the second paper of the project. Yeah.
So recently I was preparing for my final examination, and in that we have subject of drug development and clinical trials and on that. And I read that for developing a drug you need 15 years and a lot of money. And after having conversation with you, I feel that those 15 years might reduce.
We'll try. At least we will try reduce some and save some money so that at least more drugs can be evaluated. With the amount of time and amount of money required, it really can't be a pipeline where, one drug is entering at a time. It sort of has to be a funnel where you initially start with several thousands and then start narrowing it, which is okay. But then those thousands to be evaluated, we need to find resource intensive, we need to overcome the resource intensive aspects of laboratory testing and experimental methods and very complex and, sometimes technically unsuitable animal models.
In episodes of Biotech Talks, I also discussed, got to know about antimicrobial resistance and ways to deal with antimicrobial resistance. And I think this shortening of drug development might be a potential solution to deal with AMR.
Yeah, and not only for conventional antibiotics, but it's really we are past the stage where we call it a crisis,
Yeah.
We need to look at non-conventional approaches as well. And that's something that when you work in biofilms, you have the opportunity to explore. Like peptides, antimicrobial peptides, for example, or plant-based remedies, or combination treatments, and so on.
Yeah. So in your lab, you also do research to understand infection microenvironments.
Yeah.
Can you please elaborate?
So this
on
is
that?
exactly the kind of platform I talked to you about, where we have cells growing, and then we have this fourth dimension, which is the biochemical profile. So that whole thing is what we call the infection microenvironment, mimicking it as close to possible as it would be in the clinical state. So we don't study bacteria. We try not to, or if we do it, it's at a very preliminary stage. Study bacteria in isolation, like these, just floating around a tube. We try to build a microenvironment around them with cells and with the milieu, the nutrient, chemical, biochemical profile, and that is what we call the microenvironment. So where bacteria then don't attach to plastic, which is unlikely to be there at the site of the wound, but they'll attach to collagen or fibrinogen and fibronectin, and they won't be using tryptone as a source of nutrients, but they'll probably be using proteins that are present in serum, like albumin, matrix factors like fibrinogen, and so on.
Yeah, I heard that talk on YouTube. You explain how bacteria release extracellular fluids, and on that they live, and that was a really
Yeah, so the wound exudate, or what you call the extravasated wound fluid, is where the bacteria live, and it is a source of nutrients for them. So that's what we try and build in the context of wound biofilms. Yeah.
You are also trying to develop precision-based infection therapeutics.
So like this, exactly like this, where antibiotics not only is there is a stalled pipeline, but it's also sort of a one-size-fits-all, right? All gram negatives give this, all gram positives give this, mixed give this. Precision-based means you can think of a peptide, an antimicrobial peptide, that targets a specific biofilm protein or biofilm relevant structure. For example, we have work in our previous paper, which is the repository paper, where we looked at peptides that could target this enzyme in an emerging gram-positive bacterium called Corynebacterium striatum, which is multi-drug resistant, and it's being increasingly associated with ear, eye, wound, skin infections. So here we were screening using in silico approaches, screening antimicrobial peptides that could interact with the sortase C enzyme. Sortase C is needed to assemble pili in this pathogen, and pili are important for attachment and initial adhesion in biofilm formation. So this is the way we try to look at very specific antimicrobial or precision-based antimicrobial approaches that target specific features in a particular pathogen which would hopefully reduce the likelihood of emergence of resistance, would also have less toxicity, and provide a plethora of antimicrobial options. But when you do this very specific antimicrobial target combination, which is what actually our second piece of work on this repository is. But that's the kind of approach we take, for example.
Okay, so as far as I understand, like if antibiotic means you are targeting something in the bacteria, like ribosome, and then it dies.
Right.
Anti-quorum sensing means the communication that is
Right,
happening between
right.
bacteria that you target, and this is the third type, like where
So there are many types. You
Yeah.
could target enzymes, you
Yeah.
could target lipopolysaccharide,
okay,
you could target flagella. They all have to be evaluated as potential antimicrobial approaches. But when you look at precision based, means you are looking at specific agent, specific target.
okay,
And antimicrobial peptides, being so many in number and so diverse, they present an excellent library with which you can begin this kind of precision based evaluation, because we have thousands of antimicrobial peptides that we can start evaluating.
so like in antimicrobial resistance, this is what is happening. Like you do use drugs that put pressure on bacteria, and then they develop antimicrobial resistance. So is this approach also putting pressure, or it is like anti-quorum sensing molecules which do not put
Well,
that
we
much?
don't know. Resistance could be acquired uh, to any approach, but what at least to start with, and we really don't know whether resistance can be with anti-quorum sensing. Also, I don't think it's widely used in the clinic. So once it is widely used, we'll know if resistance is developing or being observed. But at least this expands the treatment options to start with. Right?
Yes.
Yeah.
So that's all questions I had. Thank you very much for
Thank
your
you.
time.
Thank you. Thank you.